Hey guys, welcome to chapter 17.4. We're continuing gene expression. We are now talking about translation. And this is one of our beefiest sections. So what I've done here is I've broken it up into sections so that we can wrap our head around it. So tRNA, we're going to be first talking about transfer RNA or tRNA. That's bugging me. And then we're going to be talking about ribosomes. And then we're going to be talking about the process of translation, which is broken up into three parts: initiation, elongation, termination. Then we're going to talk about how proteins are further modified, or polypeptide chains are further modified into proteins. And then we're going to talk once again about ribosomes, but where they are in the cell. So this is kind of our map for the day. And let's get started. So we're going to start talking about tRNA. tRNA tends to be the unsung hero of translation. It does a lot of work, and it has this cool RNA sequence and this cool nucleotide sequence, which makes it really interesting. It contains an anticodon right down here at the bottom, and it's going to contain also an amino acid on and off. Sometimes it's going to grab onto amino acids and then it's going to transport them, T for transport those amino acids to a growing polypeptide chain, or it's going to deliver it to a ribosome. And then it's going to go back and grab another amino acid and deliver it to the ribosome. So this end is the anticodon, and this end over here is going to be the amino acid attachment. And so the anticodon is going to be matched up with a very specific codon, and that codon is going to code for a very specific amino acid. So that depends. So that's going to determine which type of tRNA is going to be used. A tRNA molecule consists of a single RNA strand, and they're about 80 nucleotides long. If it is pressed flat, it would, in its 2D shape, it would have its clover shape right here that you can see. However, because it has intermolecular forces, there are complementary base pairs, which are going to be attracted through hydrogen bonds, are going to give it a 3D structure. And it's going to have this sort of L shape. And notice that the 5' beginning and the 3' end is going to be located right near each other. And at the bottom here is the anticodon. And in certain places in the book, you're going to see it represented by this structure here. So we can see the anticodon, and this would be the amino acid attachment site. So anticodons are written in a 3', 5' direction, and that is to align properly with codons that are written in a 5', 3'. So remember, DNA, that polymerase can only write down. So it's going to be writing in a 5' to 3' direction. And because the anticodon has to complementary base pair to it, it's going to be written in the opposite. The tRNA is going to carry the amino acid to the mRNA strand that is bound to the ribosome. So here is our ribosome. Here is our mRNA, or our messenger RNA strand. And you can see our little tRNAs, and they are in the cytosol, and they're grabbing onto amino acids and bringing them to the mRNA strand. The codon on the mRNA strand is going to match up with is going to complementary base pair to a very specific anticodon, and that's going to indicate which tRNA with which amino acid is allowed to attach on to this mRNA strand. So you're going to have the wrong tRNAs sometimes trying to pair up, but it won't bond. It won't make that attachment if it's the wrong anticodon. But when you have the right anticodon, it's going to pair up, and then it's going to deliver this amino acid. It's going to connect it to this growing chain of amino acids right here, or this polypeptide chain. Now, RNA we talked about is really good at molecular recognition because it has those open base pairs, or it has those open nucleotides. So it is good for recognizing specific sequences. So there are two places where tRNA engages in molecular recognition. Let's talk about the first one. The first one is where it's going to interact with this enzyme. This enzyme is called amino-acyl tRNA synthetase. Just a mouthful. But what this enzyme does, what this synthetase does, so let's break that word down, synthetase. What is it going to do? It's going to synthesize something. It's going to put something together. So it's going to take the amino acid and attach it onto the tRNA. So you can see this little purple thing here, that's the amino acid, and it's going to attach it to a specific tRNA. So not any tRNA is going to match up with any amino acid. There has to be a specific there has going to be a specific anticodon on this tRNA that's going to allow it to attach to this amino acid. However, there are about 20 different enzymes, one for each amino acid, and it's capable of binding to all of the different tRNAs that contain the same type of amino acid. So what I mean is that GCC and GCG are two different tRNA molecules. So this right here, this tRNA molecule, there's two different ones, but they both pair up with alanine. And so therefore, both of these tRNA molecules that have GCC and GCG are going to pair up to the same synthetase. So essentially, it's the amino acids that determine which synthetase will be used, or which synthetase is going to be linking it up to a tRNA. And so the synthetase catalyzes a covalent bond between these two molecules, the tRNA and the amino acid, and with the cost of an ATP, and when the amino acid is attached to the tRNA, we say that it is charged. The second instance where tRNA is engaged with molecular recognition is the pairing of tRNA, the anticodon with the codon. So here we are, we can see the anticodon on the bottom of the tRNA molecule, AGG, and here is the codon on the mRNA strand, AUC. But notice how we have some little extra G's and the C and the U. So we call this the wobble effect. So if a single tRNA molecule, if there was a 1-to-1 ratio, or if a single tRNA existed for each mRNA codon, so then there would be about 61 tRNAs. We would say that there'd be more, but there are no stop signals. There are no codons. Sorry, there are no amino acids for stop signals. So there would be about 61 tRNAs. However, some bacteria have only 45 different tRNAs. So how is it? How is it that there's only 45 tRNAs while there are 61 codons? What happens is this last nucleotide pair here doesn't have to be exact. Sometimes it is very important that the first two line up correctly, but there's some wiggle room or wobble room for the last one. So sometimes G's can pair with C's and sometimes G's can pair with U's. There are some other nuances, but I don't want you to think too hard about it or to worry too much about it. Just know what the wobble effect is, and that it allows for fewer tRNA molecules to exist than there are codons. Now let's talk about ribosomes. We're on the second word on our list here. Ribosomes, the job of a ribosome is to couple or to pair up the tRNA and the RNA or the mRNA. And so there are thousands of ribosomes in a cell. And rRNA, so therefore rRNA is the most abundant type of RNA in the cell. And a ribosome consists of a large subunit and a small subunit, and they are composed of both proteins and rRNA. So the rRNA is going to be transcribed in the nucleus, which makes sense. That's where transcription happens because DNA only exists in the nucleus. And then that rRNA is going to be shipped off to the nucleolus where the proteins are going to be added. And then they exit through a nuclear pore and exist in the cytoplasm. So a third of the mass of a ribosome is protein. It's mostly protein, and the rest is rRNA. So in a bacterial molecule, there are 3 rRNA molecules, and in eukaryotic cells, a ribosome has 4 rRNA molecules. And the function, like we said, of a ribosome is to pair the mRNA with the tRNA. And so there's a binding site for the mRNA. The mRNA is going to lay in between the large subunit and the small subunit. So here's our large subunit. Here's our small subunit. And the mRNA message is going to sort of be squished between these two. And it's going to be read. And the tRNAs are going to exist right here. They're going to pair right here. So in the A, the P, and the E. So A is where the tRNA is going to enter the structure, and it's going to read the anticodon is going to pair with the correct codon. If it's incorrect, it'll be tossed aside, and the correct one will eventually line up. Then it gets moved over to the P site, where the bond, where the amino acid is going to bond to the growing polypeptide chain. What's going to happen is the amino acid is going to be bonded to the carboxyl end of the growing peptide. So the N side is going to be connected to the C side. So if you remember, this is the backbone of an amino acid. We have our amine group here, and we have our carboxyl here. And so the growing polypeptide chain is going to be added on to this side, and then to that side, and then to that side. So the N side is going to be added on to the C side. And then after the amino acid is added onto the chain, it's going to move into the E site, and then it's going to take off and go back into the cytoplasm. Or not that it's ever left the cytoplasm, it's going to leave the ribosome. And it's going to keep doing that until the large subunit is released from the ribosome, until the message is done. Hey guys, we are now talking about the actual process of translation. And remember that we break this down into initiation, elongation, and termination. So we are talking about initiation. So the first step in initiation is when the small ribosomal subunit is going to bind to the mRNA and the tRNA. So here we can see our mRNA strand, and it's going to bind to the small subunit and the tRNA. And so when these things come together, this is the first step of initiation. So we don't even have the large subunit yet, so don't even think about the large subunit. So in bacteria, the small subunit right here can bind to, in either order, to the tRNA. It can go in that P spot, or it can bind to the mRNA first. It doesn't matter. It just needs to start upstream of the start codon. The start codon is AUG. It codes for methionine. And so it's going to be looking for this small subunit is going to be looking for the methionine, or for the tRNA that's carrying the methionine, because on the mRNA there's going to be an AUG, or a start codon. In eukaryotes, it's a little bit different, of course, they're more complex. The tRNA must bind to the small subunit of the ribosome. So in eukaryotes, the tRNA is going to bind first to that P site. Then the complex is going to attach onto the 5' end of the mRNA strand. If you remember that that guanine cap, so it's going to be using that guanine cap so that it will be able to latch onto that, and then it's going to scan downstream. So here it's going to scan downstream until it reaches that start codon, AUG. When it reaches that start codon, the tRNA is going to bond to the start codon, or so that tRNA is going to attach, hydrogen bond to that site. So here, that's the difference between bacteria and eukaryotes. So at this point, we said that there was no large ribosomal complex. And when the last part of initiation is at the expense of one GTP, that large ribosomal complex is going to attach, and where the tRNA is in that P site. So initiation factors are going to be proteins that help to facilitate this. And when the large subunit and the small subunit and the mRNA and the tRNA are all together, then we call it the translation initiation complex. And so at this point, the start codon, AUG, and the tRNA that has methionine in it right here are sitting in the P site. The A site is open, it's ready to take on more tRNAs, and it's ready to begin elongation. And the amino acid sequence is going to start. The first thing is going to be the N-terminal. And so this will be the very beginning of the protein. And then the C is going to be where the next amino acid is attached on. So here we are in elongation. Remember that translation has 3 steps: initiation, elongation, and termination. And so here we are. And the elongation process is going to have 3 steps. So try and keep all of this in your brain and in order. So the 3 steps of elongation are called codon recognition, peptide bond formation, and translocation. So let's talk about the 3 steps of elongation. Codon recognition. So the anticodon of an incoming tRNA is going to base pair with the codon. So here we have our ribosome complex with its mRNA strand, and we have our E, our P, and our A site. And we know that our tRNA is already in that P site, and then an incoming tRNA is going to come into the A site, and only the right codon will fit. And what's going to happen is it's a GTP is going to be is going to hydrolyze, and it's going to increase the accuracy and efficiency of this step. Peptide bond formation. So here, while still in the A site, so we have our tRNA in the A site, the amino acid is going to be added on to the carboxyl N to the growing peptide chain. And then when it's attached, it's going to first attach to the growing peptide chain. Then as it moves to the P site, it's going to, the bond between the tRNA and the amino acid is going to be broken. And so this is caused by adding water. It's a hydrolysis reaction. And that is going to be our second step. Our third step is when it is translocated, or translocation. So whatever's in the P site is going to be translocated to the E site. And whatever's in the E site is going to be translocated out. And whatever's in the A site is going to be translocated to the P site. So remember that the tRNA is hydrogen bonded to the mRNA strand, so it's going to be moving along with it. So you're going to have this sort of growing out strand of mRNA coming out this side as the tRNAs attach on. And this whole set, so when an A site is ready for the next amino acid, each 3-step cycle is going to take 1/10 of a second in bacteria. So we are in the last part of translation, termination. So elongation is going to continue until a stop codon reaches the A site on the mRNA. So here we have our mRNA strand, and we have a stop codon, which is going to be UAG, UAA, or UGA. So when you have that codon on the mRNA, no tRNA is going to be able to attach onto there. But what will attach on there is a release factor, a protein called a release factor. What this is going to do, when a release factor binds to the A site, it's going to cause it's going to add a water molecule to the C end, the carboxyl end of the chain, and the tRNA essentially is going to just release that growing chain from that tRNA so that it can go into the cytosol and do what it's going to do. So after that, after the amino acid chain is released, the two subunits are going to dissociate, and that's going to cost the cell 2 GTP molecules. And our second to last word on that initial list that we were reading, or that we were sort of guiding our PowerPoint here, so post-translational modifications. So while amino acids are being assembled, the chain begins to coil. So the second that it's coming out of the ribosome, it's already starting to coil, and it does this spontaneously, meaning no energy is required, and it has to do to interaction with the primary structure. So it's the intermolecular forces in there that are going to attract one another or move the protein around. And so that primary structure, it's the genes or the gene sequence that's going to determine the primary structure. So it's going to be what determines the sequence of amino acids. So there are post-translational modifications even after that. So even after the folding. So it's going to come out, it's going to fold. However, they're going to need some amino acids are going to need sugars, some are going to require lipids, some are going to require phosphate groups. Also, some amino acids are going to be cleaved away, and sometimes the polypeptide will be cleaved into multiple pieces. Also, if there is quaternary structure, those polypeptides will come together. So if you remember a hemoglobin, it has 4 polypeptides, and so those 4 polypeptides will come together and make that single protein at that point. We are at ribosome locations, our last section. There are 2 types of ribosomes, free and bound. So free ribosomes are going to be used, they're going to be suspended in the cytosol. They're free, they're hanging out, not attached to anything. And they're going to be synthesizing proteins that remain inside the cell. Bound ribosomes are going to be bound to the endoplasmic reticulum. They are the rough ER. And they're going to be synthesizing proteins that are going to leave the cell or stay in the endomembrane system or the endoplasmic reticulum or anywhere in that series of membranes. And so the chemical makeup of both populations of ribosomes are the same. In fact, they are the same. A ribosome always begins in the cytosol. And only if there's a specific sequence that indicates that it needs to go to the endoplasmic reticulum, then it will be taken to the endoplasmic reticulum. So there is a signal peptide will make sure that it gets brought over to the endoplasmic reticulum. The signal peptide is a sequence, and it's about 20 amino acids from the N-terminus, and it is recognized by a complex of proteins called a signal recognition particle, an SRP. The SRP is going to bind to the sequence on the protein. So remember, we're talking about the actual amino acid sequence here, not the mRNA. So it's going to be so this SRP is going to attach onto the N-terminus of or 20 amino acids in from the N-terminus. And that SRP is going to bring the ribosome over to the the ER, or the endoplasmic reticulum, and it's going to bind it to a protein that's in the endoplasmic reticulum. So the SRP is going to bind the ribosome to the endoplasmic reticulum, or to that protein. And when the polypeptide chain is released, or complete, is going to be released inside the endoplasmic reticulum. And it's going to then either leave. So if that polypeptide chain has to leave the endomembrane system, or if it has to leave the endoplasmic reticulum, it's going to be covered or surrounded in a lipid bilayer, so that when it goes to leave the cell, it's going to be able to leave because it'll be able to attach to that lipid bilayer around the cell. And so that will allow it, or it'll just stay in the endoplasmic reticulum. So certain proteins like insulin that are going to leave and be used in the body and in the bloodstream, those are going to be released into the endoplasmic reticulum. And so finally, we have our very last slide. In both bacteria and eukaryotes, a ribosome can synthesize an average-sized polypeptide in less than a minute, which is crazy if you think about all of those steps. However, if we need a greater number of polypeptides than can be synthesized in that time, we'll just have more ribosomes attach onto that mRNA. So once the start codon, so let's go back in time, and we're looking at when the ribosome is attached onto the mRNA. And once it reaches that start codon of AUG, and it starts making that polypeptide chain, and it moves on, then another ribosome can attach on and start making a protein. And so that is going to, I guess it would be working in this direction. So here's going to be your first completed protein. And then it'll work in this direction, and then this one will attach on, and then this one will attach on, and then this one will attach on. And so with multiple ribosomes, we call this a polyribosome. And that is the entire lecture here. So go over this a few times and make sure that you are asking questions, and have a great evening.